because we can characterize and we can characterize a particular drug, whether the particular molecule is a very important molecule or not. That we can actually deduce, we can actually decide that, right? And we also have the ability to manipulate that thing to develop a more potential drug that can simply go and bind to any of the target, any of the desired target. Okay, right? So now the thing is that sometimes, but the molecules that whether that particular molecule is going to work or not, that you can't know, that you don't know, that whether the molecule is going to bind to it, that particular molecule or not, right? So how would you be sure that whether the molecule is actually binding to that particular area or not? For that we have to perform the docking. So docking is actually nothing but a technique that we use to see if a ligand is binding to a target molecule or not. And it's not only about a ligand and target. We also can deduce the binding potential of two protein interaction, protein nucleic acid interaction, protein ligand interaction, ligand and nucleic acid interaction, all different kind of interaction that you can actually study. Because at the end of the day, we have to find out the atomic nitty-gritty of these particular details, right? So obviously for doing that, there are a few things that we should always remember. Okay, that we should always make sure that's happening. Okay, so that's what we're going to learn today, the tidbits of the docking part. What are the things that we should keep in mind, and then we're going to use the chimera and the Autodock Vena for our docking purpose to understand how you can dock one ligand with the particular protein molecule. Okay, so let's start. So now, as you see, the first thing that we have to remember is that docking is just not an easy job that yes, I simply use that particular molecule to see whether it is binding to it or not. And for that you can simply use any structure or any protein molecule. No. Obviously there are two entities in a particular docking. What are those? The first is actually the ligand molecule or the binding counterpart 1 and the second counterpart. Because two molecules, the docking is actually about the binding or affinity, or to see the atomic nitty-gritty or atomic details of the binding of two molecular entities, right? Now among them, generally one of the things that we use is a protein molecule. The target molecule is generally the protein molecule. Now while we design that, what we use that particular protein, we simply can't use any protein. No, we have to select a protein that is in a structured condition in which it is actually available in our cell. Okay, the same structural protein. But it's sometimes the proteins, they lack several things. What are the things that they lack? The first thing that they lack is the protonation state, right? Because I have already told you and discussed that some of the molecules, they have, or if you are structuring a protein molecule using X-ray crystallography, then sometimes there are no protons that are associated with a particular molecule. So what we do, we protonate that particular molecule in some software, generally using Pymol. I have shown you how to protonate a particular protein using Pymol, right? So now the thing is that when we use Pymol for our structure determination or for protonation, then what it does is generally generally you see that particular molecule is that you have to protonate it. Why? Because each of the protonation is actually different in protein. Why? Because simply think that the protonation state of a particular protein depends upon the chemical environment in which that particular protein is present. So if that particular protein is present in an acidic condition, it is going to have a different protonation state. If that particular molecule is present in an alkaline environment, it is going to have a different protonation state. So according to the chemical nature of the surrounding or the neighboring chemical balance, the protonation state of the particular protein depends, right? Now there are some amino acids that have multiple protonation sites. Which amino acid? First is actually the histidine molecule or the aspartic acid or the glutamic acid. They have, or lysine, they have multiple protonation sites. For example, histidine alone can actually be protonated in 3 different conformation, where it can actually have a protonation state at the epsilon nitrogen. It might also have a protonation state in the delta nitrogen, or it might be protonated at both epsilon and delta nitrogen. That means there are 3 different protonation states, and this actually depends, and this also controls the protein function because of the structure activity relationship. If a particular protein works well in acidic environment, for example, suppose some enzymes that are present in the stomach, then they are generally accustomed to be used in an acidic environment, right? So obviously acidic environment, they have certain different protonation states, obviously, right? So if you change that protonation state, now that particular histidine might also be present in the active site of that enzyme, and the protonation state might regulate the activity of that particular protein. If it is protonated, it is activated. If it is unprotonated, it is inactivated. So that also depends. So obviously when you are using the particular molecule for your docking purpose, it is always advised to use the activated form. So you have to make sure what is the current protonation state of the particular molecule in natural system, like in your cell, and you have to reflect that. Otherwise the data that you are going to get is not going to be a perfect data. It is going to be a false data. Okay, you can't depend or rely upon that particular data. Understand? Now similarly, sometimes what happens, so each of the molecule, they have different rotameric states, because in some condition, some of the in a particular domain, you might find the histidine or the aspartic acid is having a different protonation state. Again, at a different domain, it might have a different protonation state because of the particular surrounding chemical nature, and it also depends upon the physiological pH and the pKa of all these residues, right? So that is why correct decoding, the correct protonation state is highly required. It is a must-have technique, or it's a must-have, what should I say, domain for you, okay, that you can easily find out. Now, sometimes, but you might say that how are we going to know that which is the correct protonation state? You can't know. And that is why 3 scientists, generally the Dunbruck, at 2010, he developed a rotameric library. What is that? The rotameric library actually contains all possible protonation state of that particular molecule. Okay. All possible rotameric state. Okay. So if you are modeling a particular protein, then hardly you are not going to know whether that particular molecule will have which protonation state. So you have to use that particular library, and that library will assess the entire protein structure and give you a accurate estimation of your rotameric state. Okay, that is one of the way out, and we're going to learn how to use those things today. Okay, so that's the first selection of the particular or perfect rotameric state or the protonation state. Now, is this part clear? Okay, so I think it's clear. So now, okay, perfect. So now to the second point. Now the next important point is selection of the active site water molecules. Now in this respect, we have to talk about a number of things. The first one is the water molecule. Why should there be water molecule? Because the proteins, the ligands, they are generally present in our body inside the cell, and the cell is composed mostly of water. So generally water molecules are closely associated with a particular macromolecule, depending upon the polarity. If that particular protein is highly polar molecule, And if the particular domain is high, now generally a protein molecule, the topological surface of a protein molecule is generally a polar molecule, right? Otherwise it wouldn't be stable in that particular respect, right? Now sometime what happens, so that's the role of water molecule. Okay, next is that the active site. Now what is active site water molecule? So for that we have to determine and define the active site. Now we all know what is active site, right? What is active site? What is an active site? You can write down the answer. What is an active site? We say that it's an active site of an enzyme. So what is an active site? Come on, this is a very easy question. Yes, the substrate binds to the active site of an enzyme. Absolutely right. A part of the enzyme which actually works or which actually controls or have the activity of that, or which produces or provides the activity of the particular molecule is the active site. Now, generally what happens, we sometimes need to, when we define or devise an inhibitor for an enzyme, suppose you have a particular enzyme which is highly expressed. So obviously you want to destabilize or inhibit that particular enzyme, right? Because due to the overproduction of that particular enzyme, you have that particular disease, right? So we have to change that particular molecule, right? Develop an inhibitor. Now when we develop the inhibitor, or when we are developing the inhibitor is bound to bind to that particular active site, so that the active site can't work, it can't bind the particular substrate, and it can't produce the substrate, or it can't convert the substrate to product, right? So now the thing is, when we perform the SBDD model, the structure based drug designing, we take into account the active site of an enzyme. We calculate the volume of the enzyme, the volume of the active site, and we see the length, breadth, and the total topological surface of the particular active site, so that we can design a ligand that might fit on that particular active site, just like a jigsaw puzzle. But now the thing is, these ligand binding sites are literally a site for the protein or the product or substrate to bind. Now there are some water molecules that can simply sweep inside those particular active site and can make or form a hydrogen bond with the residues that are present on that particular area. Specifically, if it is rich in lysine or other amino acid which will participate in hydrogen bond, then the water molecule H2O, the oxygen, can obviously perform and form a hydrogen bond with that active site like residues. So now when you design a ligand, you don't know whether there are any active site water molecules present inside that particular region or not. So suppose there is a lysine residue and you devise a ligand that can bind to that particular lysine residue. But you see, and it is binding perfectly. But once you treat that particular drug, you see it is not binding with that particular protein. You are now shocked why it is not binding? Because the ligand also binds to lysine residue. But originally there are a few water molecules that are inside that particular active site which is already bonded to that particular lysine residue. And if that happens, then obviously the ligand is not going to go and bind to that particular lysine residue, right? So that is the problem. You have to take care of the active site water molecules. You have to determine whether there are any water molecules that are present inside the active site or not. And the other thing is that if there are water molecules present, then they obviously reduce the total volume of that active site. So once the ligand, now you have developed the ligand that has a complementary shape to the particular active site. So if that volume is less, can the ligand go and bind to that particular active site? Can it sweep inside the active site? It can't, because it is not fitting. For example, suppose you have a small bag and you want to fit a 42-inch TV inside your bag. You can't do it because your bag is really small. It's a 16-inch bag. Right? A 16-inch bag is not going to harvest or harbor a 42-inch TV. It can't happen, right? So that is the problem. So you have to consider the active site water molecules. And few of these water molecules are really important because they form some key hydrogen bonds. And these hydrogen bonds, they obviously enhance the ligand binding if used properly. If you are unable to use the water molecule hydrogen bond, then obviously you are going to feel that it's binding to lysine. You want your ligand to bind to lysine, but you have to consider that particular water molecule, and then you see if it is interacting with any residue, then you might find that yes, there is a particular water molecule that I have to consider, and you have to use that particular water molecule. Correct. So that is one of the way of using or selecting these important active site water molecules. Correct. Now the thing is that the next thing that we have to take care of is what? Is to understand and predict the pose of the crystal structure. Now when I say that predict the pose of crystal structure, what do I mean? Now you see the molecular crystal structure that you are using, you are not sure whether that particular molecule or ligand is having accurate structure or not, whether that particular structure is present in the body or not. You don't know it. Why don't you know it? Because there is no way of knowing it. Right. So what you have to do? You have to see whether there are any other structures or not. Sometimes what happens, there are multiple crystal structures or multiple salt structure of a protein for a single molecule. Suppose if you type in VEGF, you are going to find a number of different crystal structure of VEGF. Not only crystal, you are going to get a crystal structure, an enema structure, a different kind of structures are present for the particular VEGF. Now all of the structures are more or less similar, but there are minute modifications. Why? Because of the protein conformations. As I have already told you, proteins have multiple conformations, okay? And inside your body, according to the chemical nature or according to the physiological pH or the pK of that particular surrounding environment, the protein changes its conformation. Now this conformation change is not a huge change. It's not that it is going to be drastically different from the original structure. No, not that. A little different, but there are minute differences. Now sometimes when you design a ligand for a particular protein, then generally we have a very stringent molecule. We devise a molecule that is going to bind to this particular structured area, and it is only going to bind to this particular structure only, so that so as to ensure there is a decrease in off-target effect, because we don't want off-target effect, right? We don't want off-target effect. So we want to make sure that our ligand is highly selective in nature. It is only binding to that particular protein with which it should bind to, right? So if you have devised a ligand according to a particular protein, but suppose that particular protein conformation is a not stable conformation in our body. So when the protein reaches or the ligand reaches our body, it's going to find that among 100% of the protein, only 10% of that protein has that particular pose. So it is going to bind to the 10% of the particular molecule, but it is not going to bind to the 90% of the protein molecules because they have a different pose. So correct identification of the pose is really important. So you have to have a very great in-depth knowledge that which structure is possible or plausible in which pK, and in your condition what should be the blood pH of that particular area where the protein is present, in which pH it is going to have which pose, and selection of this particular pose is really important. Not only for the protein, but this is similar for the case of ligand also, because a ligand, it might assume a diverse role, it might assume a different kind of interaction status, a different kind of position, a different kind of pose, but only one or two of that particular pose is going to bind to a particular protein, not different kind of poses, right? So for that we have to understand which is the correct pose of the protein and for the ligand. So, and the other thing is to, now since all these what should I say, that this protein structure prediction or this docking or ligand designing is highly structure dependent and it's a computational part, you have to, the first thing is that you have to understand the correct pose, and the second thing is that you have to understand and make sure that yes, this is the particular pose in which the protein is or the ligand is bind to the particular protein, but this is also binding to it accurately, meaning you have to understand that whether your molecule or the docking server or the software is correct or not, because not all, there are multiple docking softwares, there are multiple docking servers. Which one of them are you going to use? You have to use the software or the server which you know is absolutely perfect. But how are you going to know whether your docking algorithm is perfect or not? How are you going to know? So for that, we have to devise a plan. What's the plan? The plan is to deduce or devise or design some decoys. Now the question comes, what is decoys? Now see, the ligand that you are sure that binds to your target protein is actually known as the active compound. Okay, that's the actives. When I say see the actives, decoys from actives, the actives means the ligand that is going to bind to your protein, and after binding to it, it is going to show the desired role. It is going to show the desired option that I want the protein inhibition. So yes, this ligand is binding to the protein and is showing inhibition of that particular protein activity. That is what we want. So now we are going to develop a decoy. Decoy means a particular molecule which is structurally similar, to your molecule or the active molecule, but there are certain differences that prevents that particular ligand to bind to your protein, and even if it binds, it is not going to be active. So in this way you can deduce whether the molecule or the algorithm is correct or not. How? Because the decoy that you devise, we are sure that this decoy is not going to bind to it and the active is going to bind to it. But if you see that the algorithm is also predicting that the decoy is binding to it with high precision, then you know that the ligand or the algorithm is a false algorithm. It is not working correctly. For example, suppose you want to go and show your hand to some Jyotishi and or to some Purohit who is going to simply see your hand and is going to predict your future. So how to distinguish between those truths and the false? I don't know whether there are any person who can actually predict your future or not. But if you simply go to someone and say that simply tell any event from my past, and if that particular person can correctly identify and deduce that yes, this is the thing that happened to with you when you are something very personal event to you that no one should know, and you know that that particular person is able to predict it, so you know that yes, that's a correct person. So like that, you have to design and devise few molecules that you know is 100% going to not bind to your molecule, though it is structurally similar. Now a docking algorithm, if it is 100% correct, it is only going to allow the binding of the active molecule with your target protein. It is not going to allow the binding of the decoy. So now the question is, how are we going to design or develop decoy? How I am going to know that yes, this decoy is perfect, this is the decoy? Am I going to know? So for that, that's what we need to understand. Okay, just give me a minute. So let's see. So this is the link. Okay, I'm going to define that must understand what is DUDDY. So this particular server, the DUDDY, is actually a decoy generator. If you simply show or upload your active molecule, it is going to scan the active molecule and it is going to understand and find out different cheminformatic properties like the molecular weight, the partition coefficient, the number of hydrogen bond donor, acceptor, all these things. Okay, and it is going to use and keep all these things similar, because if the structure is not similar, if the pharmacophoric properties are not similar, then that's a different molecule, right? So you have to make sure that the pharmacophoric properties are there, but it is not an active molecule because there are certain changes. Because in a particular position where you should have a hydrogen bond donor, there is a hydrogen bond acceptor at a particular place. Okay. So like that, it's going to enhance similar properties but a little different. Okay. So it's going to understand and deduce all the cheminformatic properties like that, and it's going to show you the or develop and generate the decoys. Now if you see that these decoys are also binding to your target protein, then you know that this particular algorithm is not working properly, and you have to use this with multiple algorithms. Okay, and if you see that all of the algorithms are not working properly, then you know that the algorithm is actually not a good algorithm and you have to change the algorithm. Okay, so let's see how this particular thing works. So this is the DUDDY. That is really easy to use. How to use? First, all you have to do is simply upload your email ID. Okay, so once done, it can send you the email results. And next you have to input the smiles. Now in the previous class I have already shown you how to generate the smiles from the chem sketch, right? So you can simply use that particular smile and paste it here. Now since I don't have a particular smile, I'm going to use this particular smile over here and paste it. And once you do this, and after that simply click on generate decoy. Simply click on generate decoy. Okay. If you simply click in generate decoy, then it's going to start generating decoys according to the smile information that you have put it up, and then it is going to please attempt humanity again. So it doesn't believe that I am a human. Oh, you have to write down here something. Human test, how are you doing? Fine. And then you have to upload the smile. So let me select this particular smile over here. And then simply click on generate decoys, and that's all. That's all. It's going to understand and submit, decode the pharmacophoric properties of this particular molecule, and then send you a list so that you can simply use that particular list to generate the decoys and then use a particular list for your to understand your docking abilities. Okay, is this part clear? Okay, fantastic. Now the next thing that you have to keep in mind is that, see, remember that when we have developed that protein model, then we could actually show you that after, remember that when I discussed the principle of protein structure modeling, I told you that there are 3 different steps, and what are the first step was to understand the backbone or develop the backbone. Next was to don't remember it. What was the third step? The third step was submission of the side chain to see how the side chains are working. Why side chains? Because the side chains of a particular protein molecules are projected differently. And why are the side chains important? Because side chains are the one which makes interaction with the ligands. Right? Why? Because the side chain that you have, once you develop the backbone, there are multiple side chains for a particular protein. Now these side chains, they can either project inward or they can project outward. Okay. And the side chains, they are highly flexible. Not all molecules. There are few amino acids that are highly flexible side chain. And why are they a problem? Because the more flexible they are, the more less they are going to be present at a particular position. So they are going to flutter a lot. It is going to flutter a lot because at this point or at this millisecond it is at x1, y1, z1. At the next millisecond it is going to have a different coordinate of x2, y2, z2. Similarly, after that particular second it is going to have a different coordinate like x2, y3, z3, like that. It is going to have multiple position. Now, if there are any amino acids that have a very stable side chain, then it is not going to flutter a lot. It is going to have a unique or a stable position. Now, why this is important? Because the ligand is going to make connection with the side chains. Now, if one of the side chain is highly unstable, then it is going to participate in a bond. Again at the next section it might break up the bond. Again at the next section it might participate in the bond. Again at the next section it might break up the bond. So there will be constant making and breaking of the bond. So in a way the interaction is not going to be highly stable. Whereas if you allow your ligand to bind to a particular side chain of an amino acid that is highly stable, so then you know that your molecule or the interaction that your ligand is going to have with a particular side chain or that amino acid is going to be highly stable. It is not going to make up and break up, right? So that is the issue. That is why we intend to find that we have to know whether the side chain flexibility is too much high or not. Now there is a list. You can simply see the list. The lysine and arginine are the 2 amino acids that have the highest flexibility. But does it mean that you can't use any other, you can never use lysine and arginine as a docking point? No, that's not the proper thing. You can obviously use, but it is better to shift your focus to some other amino acid that is not some, that doesn't have a dynamic side chain. Correct? Now suppose, on the other hand, sometimes what happens, you know, that you have an active site of an enzyme and there are amino acids that lines up the active site. What does that mean? That means that active site has some amino acid. Now that amino acid might have, or that protein might have amino acid that is having side chains projected inside the active site. And if that happens, what is going to happen? then again the same thing. It is not going to have a huge volume. There will be volumetric reduction and the ligand will fail to enter that docking or that active site. Why? Because the side chain is projecting inside the active site or active region, right? So at that particular location you have to change and reposition the amino acid, because never originally in wild type conformation the active sites are never occupied by the side chains of amino acid. It's never. But obviously sometimes due to the, if specifically if you are modeling the protein or if you have downloaded a particular structure, it might have this different conformations. So it is your duty to cross check and see whether it is not blocking up the binding region, because if it is blocking up the binding region, then of course your ligand is not going to work, right? So that is why these are the few things that we have to see, we have to maintain, we have to make sure that is happening. Is this part clear? Fantastic. Now the next thing is that there is also a problem with the ensemble docking. And what is that? The thing is that when we use a crystal structure, then generally the crystal structure is not, as I've told you, not the average structure, it's a specific structure. So for docking, there is one way called the ensemble docking, and there are two distinct classes of ensemble docking. And what is ensemble docking? At that particular point, as I have told you, there are multiple conformations of a particular protein. You can use all this conformation to see, whether your molecule is binding to that particular protein or not, whether your molecule is actually is binding to each of the conformations or not. So, okay, so sometimes what happens, using a particular model, different pose of that particular protein is actually hypothesized, and using that particular hypothesis they simply use the docking site like that, and they use that particular ligand and to see whether it is able to bind to multiple conformations or not. That's one of the way. And what is the other way? The other way is to take the average of all the different conformers, to see, take the average of all different conformers, and then to see whether they are able to bind to a particular region or not. Because if you see, if it is binding that to the particular region or to a particular protein, then it is binding to it. Because if you check the multiple conformations, then it is going to take a lot of time, because the protein might have 10 different conformations. So you actually have to perform 10 different types of docking. You have to perform 10 docking experiments to get the idea that yes, it is binding to that particular protein and it is binding correctly. Understand? So this is one of the things that we should actually reject, because that takes a lot of time. But if you have time, you can use that technique. So the second technique is generally preferred, where you simply use the average structure of all the proteins and use that average structure as your docking site, okay, as your docking method. Now the final problem that generally the docking has is actually to say or to deduce which is the correct docking pose, because the ligand, as I've told you, might bind might have different binding sites, it might have different binding pose. But which pose are you going to take? Because originally what is happening inside the cell, that is really difficult to visualize. You can never know how the ligand is binding to it, right? So you obviously have to predict. You have to make some what should I say? You have to make some so what you have to do? So for that reason, the docking servers have actually used the docking server they have already generated or developed a particular scoring mechanism. A scoring mechanism that scores which is the best docking pose using the binding energy. Now this current docking pose or this scoring mechanism is highly important. Because every docking site, every docking program, it is going to give you multiple options like 10, at least 10 different docking poses. But sometimes it also gives more than that, like 500, 600 docking poses. So which of the thing or which of the docking poses are you going to collect or see? The one which have the least binding energy. That means one which have the lowest delta G value. Why should we take the one which have lowest delta G value? Why should we take a molecule that has lowest delta G value? Answer. Oh, Shweta, it generally takes it doesn't take a lot of time. Okay, it doesn't take a lot of time. It generally takes a few minutes or 10 minutes, 15 minutes to give you the result. But can you tell me why should we consider the lowest molecule with lowest delta G value? At initial state high, what does initial energy high? No. What does lowest delta G negative? What does this value signify? Delta G value is negative. What does this signify? Yes, high free energy. Very good. But actually free, but what does that mean? That is the scientific term you told, but what does that signify? The significance is that the lower the delta G value, the more probable that particular binding is. Low delta G value means spontaneous reaction. The lower value, the more spontaneous reaction. So if you have a lowermost delta G value for a particular pose, that means that particular interaction is highly facilitated. That is the first, or that is what is going to happen, and then the later things can happen, but they are going to happen later. That's the first that is going to happen. Okay, understand? So that means the lowest. So this is the scoring mechanism. It calculates at the delta G value. But as you know, the delta, there are multiple delta G value. The delta G value is different. For means in a particular reaction, you have three entities. What are those? First, you have a receptor, you have a ligand, and you have a receptor ligand complex. Okay. So how do you use, or how do they use this particular scoring mechanism? They use MMPBSA and MMGBSA. And what is that? This is the molecular mechanics Poisson-Boltzmann surface area, molecular mechanics Gibbs-Bond surface area. Because it does not only takes into account the delta G value of this particular, but also other different factors. So first let me tell you about this particular thing. What it does. The MM, as you know, it uses the molecular mechanic energy of both the solvent. It uses the polar solvation terms as well as the non-polar solvation terms. And along with it, it also calculates the total entropy of the system. And obviously if the entropy increases, that means in any condition the entropy is generally entropy increment is generally facilitated. Okay, and how do they do that? First they calculate the Gibbs free energy or the delta G value of the complex, then they calculate the delta G value of the ligand, then it calculates the delta G value of the receptor, and finally it subtracts the delta G value of receptor and ligand from the complex to get the binding energy, and what is left of it is the binding energy. That's what it actually contains. But not only that, as I have told you, it uses a multiple different methods. What are the terms that it uses? It uses the, as I've told you already, the non-polar and polar terms, that is the delta E solvation energy. Next it uses the delta S, the entropy term, along with the temperature, and then it now, so what are these things? So first you have the molecular mechanics. So how are molecular mechanics formed? The molecular mechanics uses a different number of physical and chemical interaction. And what are the things? For physical, you have the chemical bond, the angles between that, and the torsion terms, that is the EBAT. And next it uses the it calculates the van der Waal interaction between the various protons, various residues, and next it calculates the Coulombic interactions. Okay, so it is not only taking into account a single kind of molecular interaction, but it is also taking into account a number of different methods like the van der Waal interaction, the Coulombic interaction, the polar solvation energy, the non-polar solvation energy, and along with it, it uses the T delta S method to finally calculate the delta G value. So it actually takes into account a lot of different energies, a lot of different interactions. Now the algorithm that uses this molecular mechanics, they give a more perfect method, or they give a more precise score rather than the molecules or the algorithm that do not uses the MMPBS or MMGBS. There are algorithms that uses only the delta G value, not the in detail terms. And if you can't calculate, or if you don't use the in detail terms, then that particular thing is not going to be highly authentic. Okay, so that is what we are going to first see and we're going to use. Okay, and next is obviously to use finally what you have to do while you develop this particular virtual screen. When you are performing the virtual screen is also a part of the docking, right? It uses the docking mechanism to understand or perform the virtual screening. Now obviously when you perform the virtual screening, to make sure that you have all the perfect things, all the interactions are happening perfectly, or that you have to include the different kind of pharmacophoric parameters. And what are the things? Like the hydrophobic moieties. So if you are using a particular, suppose you have a ligand, for example, suppose today you want to develop a medicine other than paracetamol, which can have a more potential to reduce the fever, which might have more potential to reduce the fever. Okay, so where are you going to start? You obviously have to start with the basic structure, the most well-known structure, that is the paracetamol, and you have to build upon that particular paracetamol structure to understand and answer your questions. So how are you going to do that? You are going to take all the pharmacophoric parameters from that particular thing, and you are going to solve the structure as it is. So how are you going to do that? So let's see how we are going to use the docking. So for this class we are going to use the chimera. Okay, for our docking purpose we are going to use chimera. Now the chimera hasn't opened. The chimera. Okay, open up the chimera, and let me know, you also have to use the Autodoc Vina. You have to install Autodoc Vina in your computer. So how to do that? I am just simply showing you. So this is the Autodoc Vina. This is the download option. From here you can simply download the Autodoc Vina. Okay, the Windows version is there. I'm sending you the link also, and simply install it in your computer. But make sure you know where you have installed the Autodoc Vina, because once you download the structure, you all know how to install a particular molecule. But remember where you are installing it, because we are going to require the exe file. Why do you want it? I'm going to discuss that later. Okay. So this is, once you open up the Chimera, this is what you have. Okay. Simply take a 5-minute break. Okay, I'm going to start Autodoc Vina again. So it is 7:54. We're going to start the class at 8 o'clock. Okay, simply open up the Chimera. I'm going to go and drink up water. Okay. So I'll take a break of 5 minutes, 6 minutes. I'm going to start at 8 o'clock sharp. Okay. Hi, so I'm back again. That's it. So see, once you open the Chimera, first what you have to do, we have to upload our molecules and the protein structure. So how to upload a molecule? Now see, there is an option called tools. Now everyone take a look at my screen first. Okay, first take a look into my screen first, and then later try it on your own. I'm going to give you some time to try that on your own. Okay, first take a look into my screen first, and later obviously I'm going to send you the particular application. You can still use it. So as you click on tools, the first option that you're going to get is that structure editing. Okay, structure editing. Now just a minute. Yes, so go to tools and then you have a structure editing, and under structure editing there is an option called build structure. Okay, so click on it. So first what you're going to do, we're going to upload our molecule that we have designed. Now this particular software also allows you to design your own ligand, but not in ChemSketch. You can design it here, but it's a tough job. Why? Because see, first what you have to place atom, and you have to make sure you have to give the XYZ coordinate, and by that you're going to use atom by atom connection to define or design your ligand. That's a very tough job. So what to do? We're going to click on this smile string and paste our smile. Remember, I have already told you how to have that smile string, how to get the smile string in ChemSketch. All you have to do is design a molecule, copy the smile notation, that's all. So what we are going to do, we are going to simply, suppose I am using this particular as a smile notice. Okay, suppose that this is the molecule that I have designed. Okay, so you have to simply paste the smiles notation here. You have to simply paste it. Okay, Control V, you have to simply paste it. Once it is done, click on apply. And once you click on apply, that particular molecule will be automatically built in the software and is going to be shown in your molecule. So this is the smile notation of that particular chemistry, the chemical that I have designed, suppose. So now this is the molecule that we have, perfect. But we have to now prepare the structure. Why? Because we have to ascertain the protonation state, because sometimes that hydrogens are not present there. Next we have to ascertain the charge of the molecule. So first let's see how to prepare the structure. Again we have to go to the tools, and under structure editing, see there is an option called doc prep. Tools, structure editing, doc prep. Click on doc prep. So once you click on doc prep, see, first you have is this smile notation, the smile. So you have selected it says the molecule to prep, the only molecule that you have your smile notation. So select that molecule. Yes, this is the molecule. And now see, there are multiple options here now. Delete non-complex ions. So I'm not going to discuss this right now. I will discuss this later. Once you do it, next click on OK. That's all. Now it is going to automatically add the hydrogens to your molecule. Okay. So say add hydrogens for this particular structure. So add hydrogens to the smile, and then that's OK. Click on OK. That's all. Now it is going to ask you to assign the charge to your molecule. Now, as I have told you, sometimes the molecule that you have, it might have a charged condition. Why? Because as I have told you, the charge or the protonation state of a particular molecule depends upon the environment in which it is present. So if you intend your molecule to work in the stomach, then it is going to be a pH, low pH region. So it might have a different charge like that. And next you are going to use the normal force field. The force field that we have is the FF14SB. Now there are multiple force fields like that. We have a standard residue force field of FF14SB, FF99SB, and all these are older force field. The 99SB actually supported the use of nuclear DNA and the protein, but it didn't support the use of RNA. So currently now we have the amber FF14SB, which supports the organic molecule like protein, DNA, RNA, all those things. And next is going to ask you to use the method by which you can perform the docking. So obviously we're going to use the Gastiger method. We're going to use the Gastiger method for our charge preparation, because for small molecule generally you use Gastiger method, And for large molecule like protein and all, we use the M1 BCC. So once it is done, simply click on OK. And it's going to ask you the total charge of your molecule. You can change the total charge of your molecule from this particular option here. Now generally you can keep it at +1 or you can simply keep it at neutral. Now generally I'm going to keep the molecule as a neutral charge, or it actually depends upon your molecule what charge you are going to keep. Okay, so select that particular charge and then select the gas trigger and then click on OK. Now it is going to so now see the charge has been added. Now it says check here, it says the dock prep finished. So dock preparation for this particular small molecule or ligand is finished. Now it is going to ask you to save your molecule. So let's save a molecule to a particular domain. So suppose I want to save this under this particular section, under Nidhi Shubhra and something like that. So suppose I'm going to give it this particular molecular name. So let's give its name as BI ligand, post bioinformatics ligand. Okay, and then we're going to add a mol2 suffix. So if nothing is given, you can give a PDB or you can use mol2, doesn't matter. Okay, and after that give it a name, select the domain where you want to save it. Now remember, while you do it, this particular molecule, you can't upload or select a molecule as you do in Windows. Okay, for example, suppose if you have to save it in another drive, you have to select the drive number from here, and then according to that you can simply select the molecule or change the molecule name and the space where you want to save it. Okay, so dock prep, so I did a BP and then click on save. So the particular molecule is now saved. So now we have to upload so my ligand is now prepared, my ligand is perfect. Now what we have to do, we have to upload our protein or the target molecule, right? So again, let's go to file and then click on open, because you have to upload our ligand molecule. Now you have to select your ligand. So currently you can change the drive from here. Okay, so I have already a particular molecule. Suppose this particular molecule. Okay, so see, this is receptor. No, let me go to a different. Okay, suppose I have this particular protein over here, NSP4 ab initio model. This is a particular protein. Suppose I have selected the particular protein and click on open. And once you click on open, see this particular protein molecule. Now you have this particular protein. This has been opened. Now again, I have to perform all the same job. Now I have to prepare the protein molecule for docking. What to do? Again, go to tools, then to structure analysis or structure editing. Under structure editing, you get the option called dock prep. Again, click it. Now you see, since there are two molecules in this particular option here, it says the molecule to prep, two option. Obviously, I'm going to select the protein molecule now. Now see, these are going to make sense. So now it is asking what to do. It says that for chosen molecule, do the following. Delete the solvent. If there are any water molecules or solvent molecules that are associated with this particular protein, it is going to get deleted, because I don't want my ligand to bind to I don't want to include the water molecules. It's simple as that. As simple as that. If you want to include, unselect that particular thing. Next, it says that change this particular things. What? If you have a selenomethionine, change it to methionine. If you have bromo-UMP, change it to UMP. If you have methyl-sinyl DUMP, change it to UMP. So all these things are changing. Now you see, there is an option called incomplete side chain. If you have certain side chains that are really incomplete, or that you have failed to ascertain, or people have failed to assign that particular side chain, then you can replace that particular incompleteness. You can complete that thing using the Rotamaric library. Which library? See, the Dunbruck 2010 Rotamal library is generally selected by default, because that is the best library that we have. Apart from it, there is also different library, like you have Dynanomics Rotamal library, Richardson Rotamal library, or Richardson Mode Rotamal library, but generally we use the Dunbruck library. If there are any problem with the side chain, if any side chain is incomplete, then that particular Rotamaric it is going to upload that particular structure and understand all the side chain from the rotameric library, because multiple protein have multiple rotamer. It is going to check the entire protein structure and deduce a particular or perfect rotamer according to its fitting. Okay, and it is going to add hydrogen, add charge, and all those things. So let's click on OK. Now it's going to ask you to add the hydrogens to which model. So I've saved the protein model, and now you see it is going to ask the protonation state. I taught you today, right, that very different amino acids have different protonation state. If you see histidine, glutamic acid, aspartic acid, lysine, cysteine. So if you want to change your protonation state for histidine, then you can change it like that. Select histidine and use the residue name based histidine. If in your particular protein PDB file, the histidine is generally acknowledged as histidine. If it is acknowledged as HIS, then it means that the protonation state is not known. If in the PDB file his is acknowledged as HID, that means the delta nitrogen is protonated. HIE means epsilon nitrogen is protonated, and HIP means both delta and epsilon is protonated. Or you can click on specified individually and specify it. There are two histidine. If you know that the delta histidine is protonated at 22nd position and the epsilon is protonated at 47th position, then you can select it like that. Okay, you can toggle it like that, or you can again change it to glutamic acid, change it individually, or simply click on this particular thing that used according to the residue name. Okay, and after it is done, click OK. Now you see you can calculate and you can select you can understand what I am teaching, right? You can understand the rotameric library concept. You can understand this concept that I have, the protonation concept that I have studied, that I have discussed, right? Now obviously, since this is an organic molecule, I have to use a force field. Why? Because generally the proteins, in this particular case, the protein is present in a vacuum condition. But in our body, it is not present in a vacuum condition. There are chloride ions, there are water molecules, there are multiple ions and a very thick region which everyone is actually interacting with one another. And that's how you have a very crowded environment. That crowded environment is mimicked by Ember FF14sp. And since it's a larger protein, I'm going to use AIM1 BCC to charge it residues, and then simply click it OK. And now once it is done, it is going to ask you to save the molecule. So once, would I save it? I would save it as VI. This is a receptor. So receptor DP means dock prep, and then simply click this save relative to model. Save relative to model the protein main model, because now the protein has changed. It has the charges are assigned and the molecule is in a proper shape condition, right? Okay, so to save it relative to the earlier model, and then click on save. So once it is saved, now both my protein and my ligand is prepared, right? Now we are going to proceed to docking. Clear? Okay, wait a minute now. Okay, so now once you have this two prepared structure, now we can move forward to the docking part. So how to perform the docking? Again, click on tools, structure editing. Now what to do? Now we have to use the docking method, like by using the Autodock Vina, right? We remember we used the Autodock Vina. So where is it? Just a minute. Structure editing. Where is Autodock Vina? Yes, see. So okay, so you have to go to surface binding analysis. Go to tools, then surface binding analysis, and under that you have the Autodock Vina option. See, the Autodock Vina. Under surface and binding analysis, you have the option called Autodock Vina. So click it. So now this is the tricky part. Okay, take a keen interest in this particular domain. Once you select that, first you have to now this Autodock Vina is only going to come once you install the Autodock. You have to make sure that you have installed the Autodock. Okay. So now you have to select the output files location. So you have to simply click on that particular area where you want to save the structure. You have to give a file name. So bi link prot.pdb. Okay, .pdb, because if you don't give anything, it's going to add pdbqt. So click on .pdb and click on set output location. I'm saving it in under Shubhra, so I'm clicking that's the location. So now you have to make sure you have to indicate the receptor. So obviously the NSP4 is my receptor, so that's the receptor. Next you have the ligand. So obviously I have to select the smiles as my ligand. Okay, so this is my molecule that I've selected. So receptor and ligand are now done. Now is the most important part. Okay, take a very keen interest and look into it. So now the first thing that you have to do is resize search volume using. You have to select this particular thing. Now see, the next section is the receptor search volume option. What is that? This is where we select where the ligand should bind to. Now we can use a blind docking method. In blind docking, you select the entire protein structure, because if you don't know where the ligand should bind, then you have select the entire protein and you see where the ligand is generally binding. Or if you already know the docking site, if you already know that this is the active site, this is where the ligand should bind, then you have to select that particular domain only. So how to do that? First, click on this resize search volume using, and then simply type in arbitrary anything, center 555. So this is the XYZ coordinate. And now the size. So first the size will be, suppose the size is 555 angstrom, the cube. Okay. Now click on this particular blank option. Now see, as I click on it, I got a cube. So now select. How to select the cube? You have to use your second mouse button, that is the mouse wheel. You have to select the mouse wheel and click anywhere on this black screen, not over the molecule. Remember, not over this particular cube. Select anywhere on this particular using the second mouse button, because remember we have said that resize search volume using button 2. If you select button 1, then that will be button 1. Since I have selected button 2, that is the middle mouse button. So this particular thing is, we can shift this using the middle mouse button. So now we have to drag it and bring it up on the protein structure. So see, I brought it under protein. Now to increase or decrease the size of the cube, you can do it using what? We have to simply point and select the surface. So if I want to increase this particular surface, I have to select and take my pointer to this particular surface over here, select the middle mouse button, and now see, if I move it, the box is not moving, the size of the box is increasing. See, if I select anywhere on the black space, the box is moving, but if I select anywhere on the surface, that particular surface is increasing. See, I am able to increase the surface. So suppose if I want my ligand to dock over here, then I select this particular area and I allow and I ask the molecule to dock only over here. And if I want to dock over the entire protein structure, then simply make the box big enough to sorry, make sure that the box is big enough, and then you contain the entire protein structure, obviously. Okay, now you understand. Now you see the entire protein is covered. So this particularly means that the entire protein is, you are using a blind docking. But if you already know that your molecule should bind to a particular area, then what to do? Then simply search, decrease the search volume. Suppose I want my molecule to block here, to bind here, then I will select this particular domain like that, and that's all. So for this class purpose, I'm selecting that particular location only, because if you select the entire protein, the simulation time will be more. Now see, as I move the particular thing, the center, the size, the XYZ coordinate, everything of the box has changed. Okay, so simply write something arbitrary so that the box comes, right? You are now able to do it, right? So next option is the receptor option. We are going to keep everything in the default mode. Add hydrogens in chimera. Now most of the things that we have already done, but still it is going to ask you to do it, because not everyone performs the dock prep, but it's always advisable to prepare your structure before docking. So it says add into chimera true, merge charges and remove non-polar hydrogens true. Why? Because the non-polar hydrogens rarely don't take part in a protein-protein interaction or to a protein-ligand interaction. So I'm going to select or unselect the non-polar hydrogens. But if you have time, keep it as false. But obviously keeping the non-polar hydrogens is going to increase the simulation time. So for the sake of this class, I'm keeping it as false. Okay, I'm keeping it as true. Sorry, I'm keeping it as true. But okay. So these are the rest of the thing. Ignore waters, true and all. Now the ligand option, the same thing, similar thing. Merge charge and remove non-polar hydrogens. We're going to keep it as true. Merge charge and remove lone pairs. Keep it as true. Okay. Next part we have the advanced option. Now this is really important. The advanced first option is the number of binding mode. Now as you know that this particular protein can have different conformations. The ligand also have different conformation, and it can bind to it using any pose. So if you increase the number of binding modes, it is going to search for a lot of different binding modes and give you the answer. And obviously it's going to increase the time. So if you don't want the exhaustive search, decrease it. Okay. Next is the exhaustiveness of search. That means that it is going to see every nook and corner of each of the particular amino acid. So if I select a particular area, it is going to scan the entire domain of that particular space to see where it is binding with highest affinity. And if you decrease the exhaustiveness of search, it is only going to bind to a few of the location. It's not going to find extensively. Understand? And the next is the maximum energy difference. If you have two docking pose, and if two docking pose are really similar to one another, they are going to bind with a very close gap of the energy. One is going to bind with minus 6 kilocalorie per mole. The second pose is going to bind with minus 8 kilocalorie per mole. But you get another which is binding with 2 kilocalorie per mole. Now when that happens, then that particular is not going to have an energy difference, because you can't compare 2 with 18, right? You can't compare 2 with 8 because that's a huge difference. We are going to only allow the molecule which have a energy difference of 3 kilocalorie per mole, only that. Okay. And final is the executable location. That means where do you want your software to run? Now there are two options. You can use the web service to run your software. If you use the web service, then it is going to use the server. Now in this particular case, you don't have to install the Autodoc Vina. It's going to use the Autodoc Vina of this particular Opal lab. But if your internet connection is fluctuated, if there's a huge queue, then it is going to take a lot of time, or sometime if there is not a proper internet connection, it is not going to get connected and show you the result. So that is why it's better to use the local version. Install the Autodoc Vina. It's going to take only 2 or 3 minutes, and select the vina.exe. Click on browse and select the molecule where your Vina is present. For example, my Vina is present at the G drive. So I went to G. Under G, I have the molecule under program file. So see, there we have under software, sorry, the WinLab. it is not present here. Where is the molecule? I think it is present here. Yes, see the script slab. Under script slab I have the option vina.exe. So if I select the vina.exe, then it's going to use that vina.exe for your Autodoc formation. Okay, and after that simply click on OK. And after as you click on OK, see it starts the job. The docking starts, and it's going to take some time to give you the result. So now we have to wait for a minute or two. Okay, let's wait for a few minutes to see how the result comes. Then we're going to analyze the result. Okay. Once you see this running job is done, then you know that your job has finished. Okay.
Can I do this? Let's go.
See, now the job is done. Now since it's going to show you the number of different position, okay, the more area you select, the more pose it is going to happen. So you see, first it says that this is how the ligand is binding to this particular domain. Okay, how are we going to know? So this is the option. So first you see, this is the first position and this is the second position. There are only two poses for this particular molecule because the ligand area that I have selected is a really limited area, right? So the more position, the more number is going to come here. So let's see the first model. So under first model we have, this is the best model because it has a score of 13.7. Okay, but the next is the score of 15.2. So you can obviously select the result, and now let's analyze the result. So let's see the first result that we have. So first is the active molecule. So in this particular case, these atoms are active. The C22, the C33, these are actually the active molecule. And how are this molecule behaving? So see, this is how the molecule is behaving. You see, so this is the position of the molecule 1, and this is the position of molecule 2. So if you feel that this is the best position, or the molecule is having a good position like that, so this is the molecule that we are going to select. Now you have to analyze it also. How to analyze it? First you have to calculate how, if there are a number of hydrogen bonds or not. How to calculate? So there is an option called hydrogen bond. We have to click it and click on add count to entire receptor. So now the hydrogen bond parameter is coming. First we are going to understand and see the hydrogen bond in between intermodal, between the ligand and the protein, not within the protein. So first find these bonds, the intermodal hydrogen bond. Okay, the line width 1, and now the hydrogen bond constraint. Now we know that the hydrogen bond is already only possible within 4 angstrom. So we are going to select the distance 4 angstrom like that, and on next you are going to have the constant by 20 degrees. The distance, the angle bond can have there can be a perturbation of 20 degree between the CONH. Okay, not C, NO or NH bond. Okay, and after that simply click on OK, and if there are any hydrogen bond, is going to show up. See, This is the hydrogen bond. Now see that this particular molecule is able to bind to this particular protein using this 2 hydrogen bond. But what about the next stuff using this particular model? Now we have to analyze the hydrogen bond of this particular model. So in this particular model, you see there are total 2 hydrogen bond. This is forming, and in this particular case there is only a single hydrogen bond, as you see, a single hydrogen bond, and that is a hydrogen bond ligand atoms. And what is this? Is the atom where it is binding to it. Okay, so this is the single hydrogen bond. So obviously a double hydrogen bond means it is a much more better option. So we are going to select this particular protein option only, and we have to select and save this structure. How to save it? Now go to file and click on save PDB. And then we have to select the PDB. Which PDB I want to select? Now remember there were 2 PDB molecules, 3.1 and 3.2. So, and now since I am selected only the first model, I am going to select the first model over here. Okay, this is the model that I am selecting, and then I am going to click on where I have to select. So I am going to click save on this particular folder, and I have to name it. So let's name it as BI docked. This is the receptor, right? Sorry, this is a ligand. Docked ligand.pdb. Okay, and then click on save relative to this particular model, and select the model that you want to save. I have to save this particular model over here, 3.1. Click on save. That's done. Now what to do? Now again I'm going to save the protein structure. Again click on save PDB. Now select the protein structure. I have to select the protein structure. See, I have selected the protein structure, and now I'm going to rename it using docked receptor. Docked receptor. Save relative to model, this protein model that I have selected, and click on save. Once done, now I have to go back to that particular option and see where we have saved the model. So I have saved the model over here. So the name of the model was BI receptor. Okay, see, docked ligand, docked receptor. So now we have to visualize it. So obviously we're going to use PyMol for visualization, because that's a much more better option, and you all know how to visualize or use PyMol, right? So open up one model, and now again go to file and open and select the ligand, and now see, we get the idea how the ligand is binding to this particular protein molecule. Now we have to analyze the structure. How to analyze? First we're going to analyze the polar bond. Simply go to find polar contact between chains, because there are 2 different chains. Now in this particular case it is showing that it is only having a single hydrogen bond. So, and in this particular case chimera, it was saying that it is having 2 hydrogen bonds. So this is what we have to now figure out. We have to analyze. How to analyze? So first what we have to do, we have to select the protein domain. So this is the thing that you can do. I have to select the protein domain, or the region with which the protein is binding. Okay, so this is the region where the protein is binding. Not only that, it is also binding to this particular locations over here. So this is the area. So I am also selecting this particular area, and it is also participating in an hydrogen form bond with this models over here. So I am also going to select the protein models over here, which is this is the part. So I'm also going to select that particular area and then show as sticks, and now we see how they are interacting. Or simply click on it, click on show as lines and hide sticks. Okay, so now I see that yes, this is forming a hydrogen bond with this model over here, and this is, now we have to understand and analyze with which residue is it interacting, and if there are any probable hydrogen bond formation or not. You see, yes, there is a hydrogen over there, and this hydrogen and oxygen is very close spaced. So obviously they can form a hydrogen bond. These two can obviously form a hydrogen bond over here, right? Again, you see this molecule over here. This can also form a hydrogen bond over here. But how are you going to know what is the distance between them? Have I shown you how to calculate the distance in PyMol? Which software are you running? Sweta, you have to be a little bit clear. You installed Autodoc Vina or Chimera? Are you using it as I have used it using Chimera? Because if you are using as I am doing it, then it should probably it should work. Obviously it's going to work. Okay, don't worry about it. After this class ends, take a look into this particular video, and using this particular video again, try it, and it's going to work. And if it is not working, let me know. I'm going to take a look. Okay, but generally it works because there's no way of not working. And Chudipto, you also try it. Okay, and if you are also not unable to do it, then I'm going to take a look into it. Okay. Acha, have I shown you how to calculate the distance between the two molecules using PyMol? Okay, fantastic. Yes. So now we have to find and locate the distance. So how to locate the distance? So see, there is an option called wizard over here. Click on wizard, then you have an option called measurement. So click on measurement. Now see, it says that please click on the first atom. So now you have to select the atom between which you want to calculate the distance. So the first molecule is this atom. So I selected this atom over here. See, that atom has been selected. Now it says please click on the second atom. This is the second atom, and it says that there are now see the distance is given. They are separated by a distance of 1.4 angstrom. That means they are obviously going to form hydrogen bond. Now again, I'm going to select this option over here and this button over here, and see the distance between these two atoms is 3.9, and hydrogen bond is possible at over 4 angstrom. How about these two residue over here? They are sharing a distance of 2.5 angstrom only. So that means it is obviously participating in hydrogen bond too. So there are multiple hydrogen bond options like that. Okay, and if not hydrogen bond, then you have to see the molecules or the type of interactions there can be, whether it's of van der Waals interaction or the electrostatic interactions and all. And you have to analyze and see with which model is it interacting, with which residue is it interacting. Okay, so now you know which residue it is interacting and all of that particular thing. And after that, now see, once you calculate the measurement, please click on done, because please, if you don't click on done and if you simply click, suppose I selected like that, now see, it is going to calculate everything like that, and it's the computer is going to get hanged. So what you have to do? You have to click on see delete last object. Suppose you have made some mistake like that, simply click on delete last object, delete last object, delete last object, delete last object, delete last object. Now see, the whole cluster is now actually deleted. Okay, the whole cluster is now deleted. So once the distance between every atom is calculation is done, click on done, and once you click on done over here, see, now you can select any molecule over there. It's not going to calculate the distance between them. Okay, so like that you have to calculate and find out the distance and the residues. So what is this residue? For example, click on this particular residue over here and see, this is the 28th proline. So your molecule is having a hydrogen bond with 28th proline, but generally proline can't participate in a hydrogen bond. Why? Because this is the non-polar hydrogen and it rarely participates in hydrogen bond. So obviously the hydrogen bond that I was thinking is a hydrogen bond is actually not a hydrogen bond. It can't be a hydrogen bond. So this is how now, or how you analyze the dot structure. You find out with which molecule is it interacting, where it is binding, and you all know how to use PyMol, right? I have shown you how to use PyMol. So you now have to use the PyMol and this two thing to analyze your dot structure, and then you can deduce and you can say that yes, this is the interacting site, this is the binding site. Is this part clear? Fantastic. Very good. So this whole docking and this particular thing actually concludes our this entire program, the journey that we have taken, and it was really a smooth and nice journey. Okay. Yes, today is the last class that we are having, and obviously I'm going to, not the last class, I'm going to give you an assignment, and you have to complete that assignment, and only once you complete that assignment, I know that yes, you can perform this job on your own, and that's when your class ends. Okay, so assignment will be really simple. Ask about what, Shweta? OATPB21, what is this? Can you be a little bit can you explain this? About that particular transporter. Actually, OATP1 is actually one of the transporter that is present in the hepatic cells actually. Oh, acha. You mean the drug design? Yes. Actually, yes, important. Oh yeah, I haven't decided. I'm absolutely sorry. I forgot about that. Thanks for letting me know. Now, see this particular OATP2B1 is actually one of the transporter that is actually present in the hepatic cell. It is also present in the intestinal cell walls. Okay, and what it does? It actually engulfs and the drug, it actually is the partner which facilitates this drug-drug interaction. Okay, so this particular molecule, if you have designed this particular OTP-B1 and if it is inhibiting this molecule, and then you have to know what this does in drug absorption and drug-drug interaction. Have you read about this particular OATP-B21? So generally, you know what happens that this particular molecule, it actually has a role in this absorption and disposition of this wide range of molecules or drug molecules. Okay. Now there are several compounds which are substrate of this particular molecule, right? And there are some molecules that are also inhibitor for this molecule. But the thing is that this particular thing is not that well characterized, and specifically you can't use human cell wall, right? Okay, that's enough. You don't have to read a little. So let me explain it, what it does. Generally, this particular whenever you have a genetic depletion of this molecule, and it is actually associated with the decreased absorption of this particular molecule called flavostatin. Okay, and so what is the importance? That importance is that this particular molecule is one of the, if you develop an inhibitor for this molecule, that actually also is also a class of the tyrosine kinase inhibitor that can simply destroy or inhibit this particular thing. And why? Because they actually simply, can you just give me 1 minute? Just give me a minute, please. Yes. So you see that what happens generally when you take a drug, the drug molecule has to get absorbed through your system. So there are few molecules that are getting absorbed very fantastically. It is actually getting absorbed very fast. And obviously, and there are some molecules which are not getting absorbed really fast enough. So why is there a difference? Because of this transporter. This OATP1 is one of the molecules that is hypothesized to be a transporter for this xenobiotics, for these drugs. So obviously, if your molecule is a substrate for this particular OATP1, then it is going to be smoothly absorbed by the system. And if it is not a substrate, it is not going to get absorbed. So obviously, when you design a molecule, you have to make sure that your ligand actually has some structural similarity to this particular substrate of OATP2B1.
so that your molecule can get absorbed much faster way, because the faster the absorption, the more potent the molecule is, right? So to increase the molecule efficacy, this OATP 2B1 binding efficiency is actually calculated. If your molecule is substrate for OATP 2B1, it is going to get absorbed very fast, and if it is not a molecule, if it is not a substrate, then it is not going to get absorbed beyond 50%. It might have an absorption rate of 60% but not above 80%. But if it is a substrate for OATP1, then it is going to get absorbed above 95%, and that is what we want, right? So that is the thing about this OATP2B1 molecule. So is this clear, Shweta? Fantastic. Okay, so now I'm going to give you all an assignment. I'm going to mail you the assignment, but the assignment is remember what you have to do now. Now you can design a protein, right? And you have all sent me an assignment where you have particular modeled a particular protein. Now what you have to do is use that particular protein and devise a ligand that can bind to the ligand binding domain of that particular protein. Okay, and you have to calculate which position it is binding to, and use two kind of docking. In one case you have to find out the ligand. If you know the ligand binding domain, use the ligand binding domain for the ligand attachment, and the next case you are going to use a blind docking method. That means select the entire protein as the target and see where the ligand is binding. If you find the ligand, in both the case the ligand is binding to a particular location, then you know that particular location is the preferred ligand binding zone. And if it is not binding to the decoded ligand binding zone, then that is not the probable ligand binding zone. Clear? So this will be your assignment, and you have to send it to me. And if you do it, you are going to get you have to use the chimera and everything. So you have to understand and you have to know how it is happening. And obviously you have to give me the atomic nitty-gritty too, with which atom it is interacting. What are the residues with which it is interacting? What are the atoms that is participating in this interaction and all? Clear? I am going to mail you the assignment as well. And obviously, in this particular respect, obviously, if you have any problem at all, you can simply mail it to me, and I'm going to get back to you. Okay, so how was this course? I mean, yes, Shweta. Yeah. Okay. So how was this class? Did you get to learn something new?
Yes, so
Okay. Sure. No, thanks to you that you are participating, because you know there are a lot of students comes, but someone I really feel happy when someone takes an interest on this subject, because I really feel that yes, I have done my part and someone is really interested in this particular thing. So thanks to you for understanding this class and joining this class, and thank you Shudipta also. I think you are able to learn something new, because that was the ultimate goal, that you can learn and you can apply. So that's why I go on to I perform this hands-on training, so that you can actually apply this knowledge, because if you can't apply the knowledge, then there is no meaning of gathering the information, right? Because everyone can remember, everyone can memorize. The main thing is to apply this thing, so that in your jobs and all. So that's the thing. So thank you all for joining this class, and please complete the assignment, because if you complete the assignment, then I'm going to know that yes, you are able to learn everything and you can perform the job as well. Yeah. Okay. So that will be all for today, and if you have any problem and all, simply get back to me, let me know,